Why don't we get started? Thank you all for joining us on a warmish and rainy spring day. Delighted to have all of you in person and all of those who are joining online. Today's a very special day, our Dr. Bill Bussey Endowed Lectureship. And to introduce our special speaker, I have Dr. Nizar Jarjour, our Division Chief of Pulmonary and Critical Care Medicine and Allergy.
Thank you very much, Lynn, and good morning to all of you. Welcome to our special grand round, the Department of Medicine grand round honoring Dr. William Bussey. So this is for your CME, if you didn't get it yet. So many of you know Dr. Bussey very, very well, but I doubt that all of you know how wonderful and amazing accomplishment he has made through his career. Dr. Bussey is a Badger in and out. He is a through and through Wisconsinite. Went to the University of Wisconsin for his undergraduate degree in 1963, finishing, and medical school 1966, residency and fellowship. After a brief stint away, he returned on the faculty, and he remained on the faculty until today as an emeritus professor, along the way serving as our division chief for allergy for 26 years, and the Department of Medicine chair for 2005 to 2009. It is at that conclusion of his chairmanship that this endowed professorship and chair and the lectureship has been established. Dr. Bussey served as the American Academy of Asthma, Allergy, and Immunology president in 2000, and he has had numerous major contributions to research through NIH grants and guidelines for management and treatment of asthma, extending until today. Our lectureship has benefited from wonderful, accomplished superstars and rising stars in the field, and today is no exception. Dr. Matt Altman, our William Bussey Visiting Professor for 2005, is an associate professor in the Division of Allergy and Infectious Diseases, the Department of Medicine at the University of Washington. And he's an associate scientist at Benaroya Research Institute in System Biology and Immunology. By way of introduction, Dr. Altman got his bachelor's degree in mathematics with honors from Stanford in 2004, followed by MD from Harvard in 2009, and along the way, earning a master's in philosophy from Cambridge University in the UK. His medicine residency was at Brigham, and in 2014, he completed allergy fellowship at the University of Washington in Seattle. After finishing his fellowship, he remained on the faculty and rose through the rank, got promoted to Associate Professor of Medicine in 2021. He has consistently been named as top doctor in Seattle by Seattle Magazine, and was invited to provide numerous invited international presentation at major meetings and institutions. His translational research interests focus on the whole genome transcriptional profiling, network analysis, multi-omic approaches to understanding the mechanism of asthma, respiratory infection, and other allergic and immunological diseases. Matt's research effort has resulted in more than 100 high-quality peer-reviewed publications. Most importantly for our colleagues in general medicine, Matt has been a very important collaborator and contributor to the research being led by colleagues in Wisconsin, including the Childhood Asthma in Urban Setting, CAUSE, led by Dr. Dan Jackson, the Environmental Influences of Childhood Health Outcomes, ECHO, led by Dr. Jim Gurn, and previously the Inner City Asthma Consortium, led by Dr. Bussey, and the Asthmatic Inflammation and Neurosecretory Activation, which was also led by Dr. Bussey. Dr. Altman, as you can tell, has had major research contribution and experience in bioinformatics and system biology. Today, we are fortunate to have Dr. Matt Altman speaking to us on leveraging computational biology to understand molecular mechanism underpinning immunological disease. Please help me welcome Dr. Matt Altman to the podium.
Well, thank you, everyone. I've never had such a generous introduction, and honestly, I feel honored to be here. I feel honored to be here, in particular, in honor of Bill. I mean, the truth is, I'm just, I still feel like a junior scientist, and if not for Bill, I wouldn't be here, not because he invited me, but because I wouldn't be doing this work. Bill, as I'll come to in a moment, came to Seattle a decade ago, not to find me, but happened to find me. And if not for his, endless support in every way, financially, motivation, just everything, I wouldn't have. So I feel indebted, Bill, to you, and it's really an honor to be here and to speak on your behalf. And everything I'm going to show you is based off of work that he's facilitated me doing. And you can see the title. I'm going to talk about computational biology to understand immunologic diseases, and in particular asthma today. I wanted to just start with my own brief origin story because I get to hear a lot of wonderful stories from Bill. So there was this brief interlude, which we heard, and this is what I've heard about because I think Bill came to Seattle a decade ago ultimately to find us, because he had loved his time out there. He was there in the late 60s. I hope these pictures look a little familiar. This was Madigan Army Medical Center in 1968. And I think one of the things Bill does love about Seattle is the mountain, Mount Rainier. So this is what it looks like from Madigan. You can see Madigan is a stone's throw away from Mount Rainier, about 30, 40 miles. Mount Rainier in the late summer with the glacial caps, and it's about 60 miles from Seattle. So this is all a triangle. And in the 60s, Bill was at the middle of it. And my origin story starts in 2014, and I was just figuring out what I was going to do in Seattle, working at Ben Arroyo Research Institute. I'll come to that in a moment. And Bill came out, and I was fortunate enough to get to listen in on this. And he told us about asthma, and he told us about asthma in the inner city and why this is so important to study, why he's committed his life to it. And I still use his slides, as you can see I'm doing here in almost every one of my talks. And I won't give you that whole background, just to say that the urban asthma epidemic is a big issue. It affects disproportionately the most vulnerable children, in our country, and it's essential to study. So Bill gave us this history of the NIH's investment in studying this, and University of Wisconsin-Madison's investment in studying this that goes back a long time into the 90s. This was, in effect, what it looked like when he came out to Seattle. That's his Ben Arroyo Research Institute. This is Mount Rainier in the background from one of the parking structures. It's a lovely place, and it was a lovely day that Bill was out here, and he was talking about the wonderful work that the Inner City Asthma Consortium does. And this is a figure that I'll show again, but fundamentally this is a figure that's in every good, was really a landmark study. This was a trial utilizing omalizumab in urban children and showing a decrease in asthma exacerbations, in particular with these seasonal alterations in the fall and the spring, and we'll come to that in a moment. But the reason Bill was out in Seattle, and this is just to say again, I'm gonna talk about it in a minute. I use this figure in every one of my talks. Bill came out to Seattle to say, we have this beautiful clinical data. We've made these huge advancements in understanding the disease in urban settings and treatment approaches, but we need to understand the mechanisms better. And I'll come to it. There are a lot of parts of this curve to dissect. And ultimately, really, what are the mechanisms? Because the whole point of computational biology is not to study one outcome, not to study one variable like IgE or Th2 cells, but to really try to study things in a comprehensive way and discover all the underpinnings of a clinical outcome like this. What's happening in the spring, what's happening in the fall, what's happening in between. And as Bill was leaving Ben Arroyo, he said, hey, this is great. Set up this collaboration, again, not with me, but with a very senior PI. And that PI, as Bill was getting in the taxi, and this is, I think, Dan and Jim walking to the taxi as well, they were there. He said, by the way, I'm leaving next month. I'm moving to Qatar. So good luck. But there's this, you met this junior kid, and he'll take on your multi-million collaboration. Good luck. And this is me up in the window saying, I mean, I didn't hear this conversation, but I hear the story after the fact. And again, this is where I was just luck. It was luck that I found Bill and that I was able to do this. And that's my origin story. So what have we spent the last 10, 11 years doing? Well, that's the title, again, leveraging computational biology to understand the mechanisms in these beautiful clinical and translational studies. And I'm not going to read this. These are my learning objectives. I think you have them in email and elsewhere, but what I'm proposing over the next hour is we talk a little bit about the approaches we use. We talk about some of the insights that come out of these approaches, what are the novel discoveries, and ultimately, how does this help us understand therapeutics, personalized medicine, and that is what I'll get to throughout. So more specifically, just a brief outline, and I realize this is not an audience of purely pulmonologists and allergists, as I often talk to. So I want to give a little background of, baseline endotypes. How has our field advanced over the last 30 years to define, and hopefully people are familiar with the term endotype. It was coined in the field of asthma to basically say you don't just want to know what's the clinical presentation of a disease, but what is the underlying molecular mechanism that drives the different heterogeneity of disease. And that's highly relevant in asthma. But I want to then spend most of the time proposing this sort of more novel concept that I guess I've been preaching for a couple of years, which is to say it's not enough to know what's the mechanism of disease when people are well. In fact, every exacerbation probably has its own pattern of pathophysiology, of molecular pathology. So this concept of exacerbation endotypes, and I'll come to that. I'll try to convince you of that concept, that depending on the host, the environment, and what therapeutics might be in play or relevant, you can really dissect out these different mechanisms driving an exacerbation in the fall or in the spring with a virus, with pollution. And this becomes incredibly relevant to how we think about how we're going to treat this disease in the long term. Okay, so that background, first off, here I said I'm talking about immunology, and asthma is certainly an immunologic disease. This goes back to what we all learned in medical school. This is 100 years plus of medical history, and it's simply the epidemiologic observation that asthma is most often coincident with other allergic diseases, atopic dermatitis, allergic rhinitis. What we all learned is the atopic triad. And through the years, this became increasingly well understood. We can go to the 90s, the early 90s, and a group in the UK that basically for the first time showed that if you take T cells from BAL fluid, you can see that they overexpress what we now call Th2 cytokines, IL-4, IL-5, didn't have IL-13 at the time. And this is prevalent in individuals with asthma. And this sort of coined the concept of Th2 asthma. So it's a Th2 disease, or what we now call a T2 disease. I'm going to just jump decades here. So over the course of another decade, it became increasingly understood that eosinophils are a key player of disease. I show you a piece of a manuscript here by Sally Wenzel in Pittsburgh, but Bill had everything to do with this understanding, basically to say that eosinophils are elevated in the tissue, in the blood of individuals with asthma, and that's relevant to clinical phenotype. What Sally showed here was individuals with high eosinophils in the lower airway are more likely to have a history of severe exacerbation requiring intubation. So we knew about Th2 cells, we knew about eosinophils, and then the age of molecular phenotyping and endotyping came from UCSF and Prescott Woodruff. This is, again, a landmark paper in our field, but to say that if you look at what's going on in the airway epithelium of those with asthma, you can describe this Th2 signature, or IL-13 response signature of periostin, CLK1, and SERPIN-B2 overexpression. But what's critical in a study like this is, this is only in some individuals. So about half of the adults with asthma in this study showed this molecular pattern, and half by this metric look like healthy controls. This goes along with what we know of IL-5, IL-13, elevated eosinophils. So we're sort of advancing ever more into the molecular age of defining asthma. And then again, jump forward another 10 years, now we have these beautiful diagrams that exist in the New England Journal that indicate how we will treat, and I don't have time for this story today, but basically all of the biologics in asthma came through this progressive understanding. So in this, you can see things like omalizumab, which I'll talk about, targets IgE, mepolizumab, which targets IL-5, which is the cytokine that drives eosinophils into the lung, IL-4, IL-13, and so forth. And this is wonderful, but it's incomplete. And I'll allude to why it's incomplete, or I'll show why it's incomplete, because these therapies, while wonderful, have at best 50% efficacy. And we don't understand that variability. And then there's this whole alternative side of what we just poorly call T2 low asthma, which is a way of saying we don't know. There's not T2 elevation. We don't know really what's causing that, and we don't have therapeutics. So a more comprehensive systems level understanding, I would posit, is necessary if we're going to increasingly drive new therapeutics and more precise use of therapeutics. Okay, so that's the history of the baseline endotypes of asthma. So the question that sort of evolved from 2014 to a study I'm going to show you that came out in 2019 is sort of just a general sense of, are these even static? And you can ask that several ways. Are they static when people are well? And are they static when people get sick? And there was very little study of what was actually going on during exacerbations. So I'm going to argue here that T2 asthma or eosinophilic asthma is often not static and not homogeneous, and rather that there are these distinct endotypes. And I think this is actually not hard to see. If you look at simple epidemiologic data, and I'll show you two examples, it's probably not surprising. So, you may know, or if you don't, let me briefly say that asthma, you're right, it waxes and wanes. It's sort of, there are periods of time where exacerbations are highly prevalent, and other times when they're not. And the classic in children is this fall spike that occurs when kids go back to school. That's what this big green notch is. But there are winter spikes, spring spikes, and presumably there are different triggers. And we know from Jim Gurn's decades of elegant work with rhinovirus that rhinovirus is driving that fall spike, rhinovirus A and rhinovirus C. But why should rhinovirus drive asthma through the same mechanism as an allergen or a pollutant or otherwise? And so just kind of thinking back to this epidemiologic data, you would posit that maybe there are different things going on depending on the trigger. And again, this is relevant as we think about the ICATA study here, where, again, what Bill and the Inner City Asthma Group showed was that if you treat with an anti-IgE, and this is where I want to start to target different parts of this curve. I mean, you do actually have this decrease at 2 points in time, both in the fall and the spring, which is curious because omalizumab blocks the allergic antibody IgE. Why should that have an effect on rhinovirus if rhinovirus is driving these exacerbations in the fall? So, sort of the very high-level conceptual framework was, well, these are should be driven more by pollen, these more by virus. What is omalizumab actually doing that's going to specifically affect each of these? And omalizumab, again, by its basic concept, targets IgE. What's that doing in the fall? So this was followed on then, and this is still sort of predating me and before, and this is mechanistic work by Michelle Gill when she was at UT Southwestern. So the ICATA study was followed by the PROS study, which fundamentally looked at the utilization of omalizumab just during the period that kids are going back to school to see if we can specifically use an expensive biologic for a more limited time to decrease the very adverse spike that occurs in the fall. And indeed, that's what it showed. And what was the curious finding here that Michelle showed, and it's a somewhat complicated figure, but fundamentally this is looking at the plasmacytoid dendritic cell response ex vivo, how much interferon they make. And in those treated with omalizumab, who had a high increase in their interferon alpha production, they had the lowest rate of asthma exacerbations. And those who did not recover an interferon response did not. They had a high rate of exacerbations. And fundamentally, what this taught was this concept that omalizumab is doing something more than meets the eye. It's not simply targeting allergic inflammation, activation of mast cells. It's actually functioning on the antiviral response. IgE on plasmacytoid dendritic cells, when cross-linked with allergen, prevents adequate production of interferon alpha in response to rhinovirus. Presumably, this is unproven, but presumably facilitating greater viral replication, leading to asthma exacerbations. And I don't have time for the development of this story today, but we've now done a lot more with interferon and understanding a relative interferon deficiency as a cause of asthma exacerbations in some kids in some settings. And again, this is work that's actually from one of the fellows here at Wisconsin, Courtney Gabarino, where we've really developed this story further. But the take-home message was omalizumab does more than you think it does. And the same is true with any other biologic. And if we can use these biologics to probe different mechanisms driving, response or residual exacerbations will go a long way to a better understanding of treatment. Okay, so we learned an important thing looking at this curve. Autumn spikes due to virus is probably mediated in significant part by an altered antiviral interferon response. But, and this took me a while to see, Bill pointed it out to me several times, and most of us never look at this. You know, we look at the efficacy, but in fact, the other important part of a study like this, and really any asthma clinical trial, is that exacerbations are still happening, right? So despite therapy, at some continual lower rate throughout the year, these kids are still having exacerbations, and we've done nothing to answer why. So presumably it's not antiviral issues, it's not IgE, mast cell issues, so there's got to be something else, some other mechanism driving this. And I think it's important when we think about a study like this to think about, you know, what are we not seeing in the curves? This is not only the case for urban children and omalizumab, this is the case in every phase 3 study of asthma biologics. I won't give that full history, but just to say, this is mepolizumab, a study that or a drug that I'm going to talk about in detail in a moment, towards the end of my talk. The first proven efficacy of this drug in adults, the DREAM study, a phase 3 of mepolizumab versus placebo, effective, but decreased exacerbations by about 50%. So again, what's driving the residual 50%? Or dupilumab, which targets IL-4 and IL-13, same idea, decreases exacerbations, but by 50%. So these are useful drugs. They're being used in people with T2 asthma, but either half the people are not getting any better, or, you know, everybody's getting better 50%, or somewhere in the middle of there. And there's been very little to dissect that out and to understand why is there this insufficient or incomplete response. So this fundamentally, now we're getting into the data we've developed over the last 10 years, is what led to this trial, which is called the MUPPETS-1 study. MUPPETS is mechanisms underlying exacerbations prevented or persistent with immune-based therapies. But fundamentally, we wanted to profile, and we wanted to profile as comprehensively as we could, what was going on in children during their exacerbations. So the framework of the study was enrolling children who clinically you would say are T2 high. They have elevated eosinophils, they have allergen sensitization, they're exacerbation prone, meaning they have multiple exacerbations per year. And we wanted to look at them at baseline, as many others, as Prescott Woodruff and others had done, and understand, you know, what's their molecular profile at baseline. But then we wanted to get samples when they were ill. So we gave them a smartphone, had them track symptoms, and brought them in as soon as they had symptoms of an upper respiratory illness, and we got repeated measures during those illnesses. We collected blood, we collected airway, we looked at what we could, and ultimately found that upper airway transcriptome profiling provided a wealth of information. And you know, this is a number of years ago. This is all by bulk RNA sequencing. I'll talk a little bit about methods. I'm not going to get to it today, but you know, we're now doing single cell in the same setting. And there's a lot you can do with these samples if you get good longitudinal data at important time points. So the methodology, it's pretty simple. As Andy Liu, one of our collaborators, puts it, we're just collecting snot. You lavage the upper airway, you use a sinus rinse, you get a cell pellet, which is a mix of epithelium, eosinophils, immune cells, kind of all the relevant inflammatory players in the airway. We get viral information thanks to Jim Gurn. We get cell differentials thanks to Michelle Gill. We get sequencing at our core, and you know, other things I won't get into, microbiome, proteome, other things we can get from these types of samples. And fundamentally, we're looking at the airway response and alongside it, the peripheral response with blood. So as we get into this idea of big data, sorry for the change in formatting here, but you know, when you collect these samples and you do sequencing, you get a lot of data. So we're not measuring simply interferon, we're not measuring simply IgE or you know, periostin or what have you. Rather, we're trying to comprehensively profile. And I'm sure many in the audience are familiar with this type of data, but you get about 15,000 parameters, right? Every gene that's expressed in that tissue or in that sample type. We get cell quantification. The statistics behind this get tricky, right? Because you have inflated false discovery rate if you just look at every signal. So you really need some nuanced ways to dimension reduce this and to find the patterns in the data. And fundamentally what we do here, because we have a heterogeneous sample type, is we deconvolute. So we use fancy network analyses to say what genes seem to be coming from the eosinophil, what genes seem to be associated with the epithelium, the mast cell, so on and so forth. And then once we partition genes to the extent we can by cell type, we then partition them further through what's called weighted gene co-expression network analysis into these modules. And we go from about 15,000 variables to about 50. And what those modules look like, and I'll show you throughout this talk, is this. They're data discovered. So we didn't tell, from canonical information what modules to create. This is simply the structure of the data in an unsupervised manner, and we find the biology embedded within. So this is a T2 module, IL-4, IL-5, IL-13, relevant mast cell and eosinophil genes that fall out of the data. And similarly, I'll show you many others that have nothing to do with T2 inflammation. We annotate them, and then we can do things like kinetic modeling of them in different circumstances of exacerbation versus not. So that's exactly what we did. And before I show you some of those kinetic models, the first thing that came out of this data, this is an unsupervised PCA plot of illnesses, of just the exacerbation illnesses. The first thing that was pretty obvious is that if you have a virus or if you don't, these exacerbations look pretty different. So the viruses are the blue, the non-viral exacerbations are the red, and clearly in a global sense, they look pretty different. So what we chose to do fundamentally was model viral exacerbations versus viral non-exacerbations, non-viral exacerbations, and then non-viral non-exacerbations, whatever those may be. The kids are sick, they don't have a virus, they don't get an exacerbation. And we can now look across our 52 different modules and say what's relevant to each of these groups. And there are things that are definitely conserved or shared between viral and non-viral exacerbations. So this module, which fundamentally has to do with TGF-beta signaling in the airway, SMAD3 transcriptional regulator, is very markedly upregulated in both, maybe with a slightly different kinetic. But before the exacerbation is occurring, which is generally out here around day 3, 4, we see this brisk upregulation around day 0, 1, 2. That seems to precede the onset of a true exacerbation. In contrast, there are those that are very specific. So here is what looked like a viral exacerbation specific module. This had many known players like IL-33, an alarmin from the airway epithelium that in mouse work and various other studies we know to be relevant to allergic inflammation, but also a whole host of other ciliated genes, including something like CDHR3, which many may know the story that Jim Gurns put around that. This is in effect how rhinovirus C infects the cell. So if anything, this is perhaps priming for repeated infections. And then separately, we can describe what's going on. And at the time, we didn't really know what a viral negative exacerbation was, but it had its own pathways. So tissue kallikrein induction, quite curious, and aspects of IL-17, IL-23, and IL-36 inflammation that was uniquely upregulated in these viral negative exacerbations. So suddenly we have this kinetic profile of what ultimately was about 20 of the modules that seemed to, in different kids and different subgroups and different triggers, be relevant to what's underpinning these exacerbations. And we made cartoon diagrams back then saying, if you have a virus, this is what happens first. Then you see these core modules like SMAD3 TGF-beta, ultimately this excessive interferon signaling. And during the time of exacerbation, as you can see by this lower triangle, what we called effector pathways. So mucus hypersecretion pathways, further eosinophil activation, extracellular matrix, protein or gene upregulation, and things like that. And with a non-viral exacerbation, we had different entry points. So early on, we were seeing non-ciliated epithelial responses, tissue kallikrein, Z cadherin, keratinization, suggesting that these are, while they're converging to some similar molecular pathology, they really start in different places. So that was great. We've done a lot with those data. It was an observational study to say what's actually happening during different exacerbations. The next question that came of this, or the next one I'll tell you about, is we wanted to figure out what these non-viral triggers were. Again, these are urban children. They have quite distinct environments in which they're living. And we went searching. This kind of indicates we went searching for allergen. We went searching with microbiome data. And then we pulled in a lot of exposome data. So we pulled in pollution data and found that that seemed to be where the signal was. So this is now showing the kinetic profile of the air quality index matched per each individual based on where they live during the time of their illness. And we found the colors have switched. In this paper, we called viral negative exacerbations the green. But we saw this spike of elevated air quality index occurring around the time they were getting sick and kind of remaining elevated throughout the exacerbations. This is day before cold onset, and all the exacerbations occurred within this time point of sort of day 0 to 9. So that seemed to be relevant, but that's just an association. We wanted to ask, and I'll say in terms of the AQI, it's a composite of 6 different pollutants. We found the relevant drivers there to be variably ozone, NO2, or PM2.5. We could actually link these directly to the different modules. So again, this is associative, correlative, but fundamentally we found during those spikes of PM2.5, the cumulative exposure to it directly related to the relative induction of the tissue kallikrein module that was most pronounced in these viral negative exacerbations, but occurred in everyone. And in contrast, in the ozone exposure time points, which tended to be more in the summer, the PM2.5 more in the fall, that associated with higher expression of ozone. So we're starting to link together the exposure, the molecular driver, and the outcome. All right, so what I hope I've done there is convince you that we have this profiling method. We can use this to understand what's going on with different triggers. And it's certainly looking as though this idea that I've kind of after the fact postulated of different exacerbation endotypes is true, even in a population of kids we would otherwise call T2 asthma. I'll just do a brief interlude that the host is relevant here too. So we work in the Inner City and Now Cause Consortium with Carol Ober, and she's a wonderful geneticist. We look at genetics, epigenetics. I just want to show you one brief story that came out of this. And that is around mucus hypersecretion and mucus plugging. So this has been of great interest for the last decade or so. We know mucus plugging is relevant, not only in asthma, but in other pulmonary diseases as well. And in the Adult Severe Asthma Research Project, there were 2 great papers in 2018 basically showing that if you're a mucus plugger, you tend to remain a mucus plugger over time. You tend to have mucus plugs consistently over time. Finding that if you're somebody who has a high burden of mucus plugs, you tend to have lower lung function. That's what's shown on the left. And molecularly, you seem to have more MUC5AC, which is the pathophysiologic mucin relevant in mucus plugs, than you have MUC5B. And that there's a rough correlation of mucus scores in the lung to eosinophils, but these are by no means one-to-one. You can have a high mucus score in a relative absence of eosinophils and vice versa. So there was work, and we jumped into it with our data to say, well, what's going on here? And there was a UBioPred, that's a European consortium, that found for the first time that MUC5AC gene locus is a GWAS site. It's a genome-wide association site for severe asthma. And what we went on to show in our data was that based on your MUC5AC genotype, this is again in Muppets and also in our Eureka cohort, that if you have the risk allele, the T allele, at this site, you're more likely to have lower lung function. But again, what was critical is we had illnesses, and we could see that this induction of MUC5AC, so this is what's called an inducible EQTL analysis during exacerbations, you upregulate MUC5AC quite specifically if you have the risk allele. So this is less about some sort of basal overproduction of MUC5AC, but with the right genotype, you upregulate and presumably, are more prone to forming mucus plugs if you have this and if this is happening during exacerbations. So just, again, this is one of many examples, but layering on how the host and presumably, whatever environmental trigger here intersect to drive what phenotype you have. So that's, again, a brief story about host factors influencing exacerbation endotypes. But what I want to spend the rest of the time in is how does this ultimately drive what we do with individuals with asthma? How does this drive asthma therapies that exist now or that might exist into the future? So for this, we fundamentally, and as Bill put it, and I think the way he characterizes is wonderful, is basically like we can use these biologics that have been developed as a human knockout, right? We can knock out IgE, we can knock out IL-5, and we can ask, why does it work in some individuals and not in others? We can use this towards an ever-refined understanding of the pathophysiologies of these exacerbations. With this, we want to get biomarkers. We have these crude biomarkers like blood eosinophils. Can we do better? I'll show you a story of that. Can we identify what's going on in non-responders so that we can develop treatments for those individuals? So Muppets 1 was a pilot. We did Muppets 2 next. This is a randomized controlled trial. You can see the title there, but again, we're looking at embedding mechanistic data into a randomized controlled trial. So the cohort is the same concept. So children in urban settings with elevated eosinophils, with allergic sensitization generally, and with multiple exacerbations per year. So sort of the most severe asthma in the most vulnerable populations. They go on to therapy either with mepolizumab, which I mentioned before. It's a anti-IL-5 drug, so it depletes eosinophils from the blood, from tissue, and it's been shown effective, as I mentioned, at about a 50% efficacy in adults. We wanted to compare it in these children where it had not been studied to placebo and assess its relative efficacy there. So the primary outcome, like every good asthma interventional study, was the rate of exacerbations that occurred while on therapy. And we embedded within it these mechanistic studies listed below. Just like I showed you before, I'm going to focus in on the airway transcriptomic data and what it told us about the relative efficacy of this drug. So this was the primary outcome of the study published by Dr. Jackson in The Lancet 2022. And it was effective. The drug did work. I think what was most interesting to us and perhaps disappointing to GSK, who manufactures the drug, is it was not as effective as you might have anticipated. So diminished by about a quarter as opposed to about a half exacerbations in this population, but significant. And while not necessarily the most robust clinical outcome, what's really nice about data like this is it gives us the opportunity to understand failure, mechanisms of failure, and maybe even mechanisms of adverse response to drug. So that's exactly what we endeavored to do. So the question, what are the mechanisms underlying those exacerbations prevented or persistent despite anti-IL-5 therapy? So I showed you this before, right? Same idea, take the bulk nasal lavage RNA sequencing and the associated data. In fact, apply the same modules we had developed a few years earlier and see, one, are they relevant to this data set? We hope that they are. And two, what do they tell us about therapeutic responsiveness? So this was the main mechanistic figure accompanying that clinical outcome. And it's quite interesting. Now, there's a lot here, and certainly please go and read the paper if you want all the nitty-gritty. But at a global picture, what you can see is that, somewhat as expected, these eosinophil-associated modules, including the type 2 module that I've shown before, eicosanoid metabolism, other aspects of eosinophil gene expression, this is a PLS regression plot. And what that means is it's showing you the relative influence of the baseline expression of this module on outcome. So if you're on placebo in red and you have high expression of this module, you're going to do poorly throughout the course of the year. You're going to have a high rate of exacerbations. Conversely, if you go on to mepolizumab therapy, you're going to have and you have high expression of this, it doesn't influence your exacerbation rate. It has no relationship to it, which in effect means that presumably you're downregulating this, and I'll show you that in a moment. And this no longer becomes relevant once you go on therapy. However, there are other aspects of the eosinophil, namely this concept of activated eosinophils and their intersection with airway mucus hypersecretion, where there is no influence of drug. If you are overexpress this mucus activation pathway, which I mentioned a moment ago is probably at least in part genetically controlled, you're going to do poorly on or off drug. So that component of mucus hypersecretion related to eosinophil activation appears not to be relevant to the efficacy of this eosinophil depleting drug. And then most interestingly, perhaps, were this whole block of epithelial-associated pathways where you can see it's the opposite. In effect, if you overexpress these pathways, which is unfortunately common in these urban children, you may actually do worse on the drug. So you have a high rate of exacerbations if you go onto the drug. And they're not so much relevant, many of these, if you're on placebo. And again, this is something like the TGF-beta SMAD3 or some of the other epithelial modules I've alluded to. So suddenly we're getting a picture of responder, non-responder, and even prediction of who's going to do well. And the other half of this figure was to say, sure enough, once you're treated and you've been on therapy for a year, or for that matter for 8 weeks, because we had an 8-week time point as well, you see that these eosinophil modules are diminished in the airway. And in fact, these epithelial modules are elevated. So the drug is doing something, whether it's a reciprocal elevation because the eosinophils are gone, or it's a direct effect on the epithelium, still to be determined, but it's inducing some level of these airway inflammatory pathways. So very unexpected. And beyond that, and this is as yet unpublished data, but you can take it a step further. So this is a machine learning approach, effectively like a random forest, if you're familiar with that term, where we can say, let's partition our population in a cross-validated manner and ask, do these actually partition our kids into responders and non-responders? And you can do this at the gene level, you can do it at the module level, but fundamentally you can take branch points on your relative expression of one of these modules and say, okay, and I'll show you this in a more intuitive figure on the next slide. If you have high expression of this and you're on mepolizumab, that's a bad thing. If you have high expression of this and you're on mepolizumab, it's a good thing. And then there's this whole set where it doesn't matter. And what that looks like is now we can take our outcome of rate of exacerbations and see that sure enough, if you're a high expressor of this epithelial extracellular matrix production module, you'll do worse on mepolizumab than the parallel placebo. These are the kids that we wanted to target, those who had true airway tissue T2 inflammation, at least as measured by this eosinophil eicosanoid metabolism module. There's the more expected, highly robust 50% reduction in exacerbations. And then, a significant part of the population who, despite having elevated eosinophils and allergic sensitization, they didn't need the drug. They were equivalent whether on placebo or not. So we're getting an increasingly refined way of picking out those individuals who would most benefit from therapy or who really should not be on it. All right, the sort of final story I want to pull out of this that we've just published is kind of going back to what we saw in Muppets 1 is that's all great, what's going on at baseline and your baseline endotype, but what's actually driving these residual exacerbations when you're on the drug? So much like in the Muppets 1 study, we sampled during illness. So we sampled during illness when you're on placebo and when you're on mepolizumab. And that's what's shown here. Fundamentally, same idea. These children were asked to report or to let us know when they had upper respiratory illness symptoms, many of which lead to an exacerbation, to come in as quickly thereafter, day 0, 1, 2, as they start to feel sick. And we would collect samples before they exacerbated or resolved that cold and before they were on corticosteroids. So here we could compare mepolizumab versus placebo exacerbations, relate to lung function. And the hypothesis here was, well, as I sort of mentioned before, there's probably more than one mechanism. Now, if it was all T2 exacerbations and maybe just the drug's only 50% effective in reducing it, you'd expect something like this. Mepolizumab partially decreases, or in some people adequately decreases a specific pathway, be that eosinophilic inflammation or antiviral responses or whatever it may be, and the residual exacerbations are just because you have too much of that pathway. That's obviously, as probably my talk has led up to, not going to be the case. Rather, this idea that there are multiple axes of inflammation. And it may be that when you block one, the other is what's driving the exacerbation. And this can be within a single child. This is what gets tricky. A kid may exacerbate one day through one pathway with one trigger, and another day through another pathway with another trigger. And so here I'm postulating this idea of a T2 response or a TGF-beta epithelial response as two examples. But let's see what the data actually shows. So what we found was, and here we're basically showing the relative expression of different modules as I derived them previously, when your baseline in the mepolizumab group, so before you're on therapy, what it looks like when you have a mepolizumab exacerbation. So you're on drug, you have an exacerbation, a resolving illness, and what it's like at the end of the study. And you can appreciate this small difference here. This is what I showed you before, that just going on the drug over the course of a year, you do have a small increase in this epithelial pathway. You don't see that in placebo, and this is, I'm sorry, I guess this is a Mac to PC thing, but this is the same placebo baseline, placebo exacerbation, cold, and end of study. But what you can see is this is markedly induced during mepolizumab exacerbations, and to a much greater degree than what's observed in a corresponding placebo exacerbation, where it's elevated to an incremental degree. So mepolizumab exacerbation, and the same is true of many other modules, but with different patterns. So this next one, not a module I focused on before this slide, but one that was relevant in Muppets 1, this heat shock protein stress response module. I show it simply because it's equally relevant in the two. We see it upregulated in MEPO exacerbations just as well as placebo exacerbations. So there are different patterns at play, and you can see this globally if you look at a heat map. So showing the relative expression of, this is only 14 of the over 20 modules that we saw as relevant to these different exacerbations, but trying to summarize the high-level biology. You can see that the things most relevant to the mepolizumab exacerbations seem to be epithelial-derived, again, congruent with what we understood from the baseline predictors, and macrophage inflammatory pathways. Whereas, those are relevant, they're somewhat higher than colds in placebo, but the major story, or the more relevant story in placebo, had to do with the eosinophil. These modules, similarly, it's not just the exacerbation outcome, but as a continuous metric, they show a direct relationship to lung function decline during the illness. So I'm just showing you those same two modules. The higher the expression, the lower your lung function as measured by FEV1% predicted, or FEV1 FVC in each case. And then the final step, I mentioned before, the trigger matters. What we saw is that if you partition the exacerbations into viral positive or negative, you can see it's distinct. A viral negative exacerbation in mepolizumab is more so driven by several of these epithelial modules, extracellular matrix production, EGFR signaling. The SMAD3 is sort of common to each, whereas the viral positive exacerbations on mepolizumab are more driven through this alternative macrophage inflammatory pathways described in these labels. And what we ultimately did, and I'm not going to show you the figure because it's really big and complicated, but you can now apply a multidimensional approach to this. And we can only visualize things in 3 dimensions, so I show it to you in 3 dimensions. But fundamentally, you can start to define different axes of inflammation. And it's not that there are 50 or 100 or an infinite number of axes, but you can reduce it down to a handful. And it's not 1, it's not 2. Here I show you a 3-axis solution, and it may be a few more than 3, but fundamentally, depending on the host, depending on the trigger, depending on what therapy they're on, you're more prone to exacerbate through one, the other, or some combination of these. So you can have very high overexpression of these epithelial remodeling pathways and exacerbate. You can have very high expression of these eosinophil pathways and exacerbate, macrophage, or some combination therein. And of course, if we have a therapy that only affects one of them, you're only going to reduce exacerbations 25% or 50%, or depending on how relevant that pathway is to the group. So that's the story as it exists right now. And it's good, I'm about at time. So to summarize, asthma baseline endotypes. Over the last 30 years, that's what allowed us to develop the drugs we now have. It gave us, 25 to 50% efficacy in reducing exacerbations. It gave us biomarkers like blood eosinophils to select these therapeutics. What we're learning is that host and environmental factors are critical to this concept of exacerbation endotypes. What's actually occurring in the illness? How are we going to prevent those illnesses? And ultimately, I've shown you just by bulk RNA-seq, but again, through these embedded, unbiased, basically surveying everything you can survey, data generation and computational biology approaches, you're going to discover not only what you know, but entirely novel aspects of biology in, as Bill puts it, a human knockout study. And that's going to lead us towards novel therapeutics. We have therapeutics around kallikreins, we have therapeutics around interferon. Is that going to be relevant in asthma over the next decade? Uh, so these large molecular, this is what I just said, I think. These large molecular data sets being embedded in clinical trials gives us that novel insight. And ultimately, this is why, we talk about precision medicine as kind of a hyped term, but ultimately this is giving us ever precise understanding and the option for not only targeting our therapeutics, but thinking about repurposed or new therapeutics in this space. With that, I'll conclude. A lot of acknowledgments. Again, it's such an honor to be here. None of this would happen without the tireless contribution of Bill, who has mentored so many, and I feel lucky to be one of them. Dan Jackson, who's here and is now the PI alongside Jim Gurn and driving these studies forward. The support of the NIH, which is more important now than ever. We just flew in from NIH yesterday and had a wonderful meeting on exactly these topics. And we're funded and going to continue this work. So look forward to years and years of collaboration to come. My group at Benaroya in particular, well, everybody listed has worked on this at some level, but Kim, Naresh, Max, these are all bioinformaticians. If anybody's interested, I've actually mentored some of the fellows here. My lab meeting is open. If people want to learn these methods, shoot me an email. We meet every Tuesday. We talk about data and everybody involved in ICAC and cause. And with that, you have my contact. Thank you, and I'm happy to answer what questions I can.
Great. Thank you, Matt, for a great journey. Love the curling.
Yeah, right. I forgot to mention that. We do have it out in Seattle. That's what we do every year.
I actually got my first curling experience in Seattle.
Yeah.
So,
good.
I think the other thing that impressed me is the evidence of the lineage tracing of the influence of Dr. Bussey and the group, and shows how mentorship and collaborations really expand our field and are so crucial to developing the next generation and expanding our influence throughout. And so also want to thank Dr. Bussey for his support of this lectureship, to support of the research, and for investigators around the world. So this, his support enables us to bring in outstanding investigators from around the country, around the world, to share the work with us and to then be able to continue to influence others with your support as well. So I'm going to hand it over to Dr. Jajor for further presentation.
Thank you very much, Lynn. So I'm going to invite Lynn to stay and Bill to come forward for the presentation of the plaque commemorating this occasion. So come with me here, and is Anne or somebody can take a picture? So this has been a tradition for the Bussey professor to give them a plaque having their name and the shape of the map of Wisconsin. So Bill, you can. So congratulations.
Okay. There you go. Forward still.
All right, so now we can take some questions.
I'll ask the first. So I'll ask the first question. So thank you, Matt. So as you're talking about it and thinking how you're going to apply this to developing a strategy to treat patients, because what I'm hearing is for an individual patient, the appropriate therapy is going to depend on the exposure and time, et cetera. So how do you envision managing this? So like fall, we're going to give people, so.
Yeah, I mean, it's a wonderful, oh yeah, I've got the mic. It's a wonderful question and a complicated one, right? And I think my last bullet point there was this is why it's both exciting and complicated. And I'm not going to sit here and tell you I have the perfect solution. I certainly don't. But we saw already this concept, and not everything is a biologic, but even when you take a biologic like omalizumab, and this unfortunately has not come into clinical practice as perhaps it should, but using it during a brief period of time when you know that rhinovirus is relevant and interferon deficiency is relevant. So identify those kids who have a relative deficiency in their basal interferon tone, which we're now doing, treat them for a brief period with the drug of relevance during that time of year, and you're going to markedly mitigate all of the morbidity that occurs with these, or you may, with all these school time exacerbations. So that's one example. But yeah, same idea with these other findings. I mean, I don't want to suggest again that there are an infinite number of pathways and every kid exacerbates through every pathway. But if you start to get a profile of a given child, when they exacerbate, through what mechanisms, or even if you just have a baseline signature like, oh, they're high, TGF-beta SMAD3 at baseline, mepolizumab's not the solution. We need to think about perhaps that's a kid who needs omalizumab. Perhaps we're talking about other things that would affect these pathways that are non-biologic as well. So we need to test and trial these, and there are different. I'm not a clinical trialist, but of course there are a variety of clinical trial designs now that could facilitate that, right? So where you may have multiple drugs compared against one another, switch drugs depending on the molecular data and the clinical outcomes that occur, and you can start to get ever more precise. So I think a combination of these strategies is where we go.
Great.
Well, that was an excellent talk. And I think this other endotype of TGF-beta SMAD is really surprising because I usually think of that as more of a chronic remodeling type signal. So the question I have is are those patients different in some way? For example, do they have any evidence of chronic remodeling at baseline? Is their baseline lung function different?
Yeah. So it's a wonderful question and something we really want to explore. And the best way, of course, to explore that would be to have long-term data on these kids, which we may accrue. We're actually looking at this in a cohort called CREW and CANOE and other places. But it shows we basically, this is sort of cross-sectional data across 6 to 17. So we don't see this strong baseline association to lung function. Rather, as I showed, we see that it's markedly associated during illness. Now, the idea in the field is when you have repeated exacerbations, this is what leads to incremental decline in lung function over time. That's the postulate here. Whether we can prove that, it may not be in a one-year follow-up study, but maybe in some of these birth cohort or other studies. And indeed, in Eureka cohort, we have evidence of what's relevant to loss of lung function over time.
Yeah, so I'm going to follow up on Nate's question because I was fascinated by the TGF-beta also. There are a lot of therapeutics targeting TGF-beta pathway in fibrosis. Are any of them being tested in?
Yeah, so of course, I mean, the problem with TGF-beta is it's such a pleiotropic, cytokine, and and this is children. So the systemic interventions are probably not where you would want to go, but certainly what I've thought about a lot, and yet to really reach out to any pharmaceutical or anywise, but could an inhaled product here be relevant? And what's relevant? Do you want something that blocks TGF-beta 1? Do you want something that has an effect on SMAD3? There are certainly unknowns there. And some of this we need to follow up with ex vivo ALI type modeling, but I think there's potential for a role to play there. And again, keep in mind it may not be something you're on chronically, right? If this is only happening during illness, there is this concept of using things around the time of illness, be it something that might block that pathway, might augment your interferon, transiently when you're at risk. So these could be short-term therapies.
Matt, thank you for the journey. It was just really wonderful on how you can pull all this together. The question I have is taking this to the next step, preventing the disease. Can you put this together and sort of identify when the original event or events happen and then sort of prevent the disease from expressing itself, which would be the ultimate thing that we'd like to accomplish?
It's tough, right? He's always asking the most critical questions, right? And this is the direction, and remission of the disease once established and prevention of the disease in early childhood. And the short answer is we hope so. I mean, this is what, of course, the CANOE cohort that Jim leads is all about. We're basically identifying the things that are happening in the first 4 months and the first 12 months of life that seem to be driving this. And those children are not going to be 18 years old for a while, but we already have some clinical outcome data. We're relating these early life alterations in the epithelium and the immune system to what looks like early clinical disease, and that gives us the postulates of what are we going to do. I mean, we probably don't want to give everyone a biologic when they're 4 months old, but rather this points to things, whether it's, other mediators at the epithelium, antioxidant pathways. There are a lot of ideas that, of course, come up, but we need to know the molecular underpinnings early life first to get to that point.
And then are these phenotypes, endotypes replicated in adults?
Yeah, it's a good question, right? So we we have a lot of adult people here, I'm sure, and I'm adult trained. So is Bill. We study kids because it's important and because it's very prevalent there. I think the short answer is we don't fully know, but I'm sure a lot of it is. So just take, for example, the T2 biology that Prescott defined in 2008, 2009. We see this exact replication in children in the Eureka cohort. And as you start to take some of our novel data and look at it, you can see the echoes of it in some adult data as well. But nobody's done this level of profiling is the reality in adult populations, and we should. And actually, there's talk of how we may do that with collaborators here.
Well, we are at the hour. Thank you again for a wonderful journey. Thank you, Dr. Bussey, for the support of this, for all of you for your attention. I think the other lesson I took from this is, being able to use this data to help continue to support public health policy that's going to impact the health of our children, the health of our nation, our population. So thank you very much.
You're all.